My name is Shuyu, work on wheat genetics in Amarillo Center. It's my great pleasure to introduce Mr. Mustafa Cerit. He's studying for his master's degree in plant breeding and genetics with Turkey Government Scholarship. He joined Soil and Crop Science in the fall of 2019. He's co-advised by Dr. Amir Ibrahim and myself. He has finished all the required courses. He came to Amarillo to work on his thesis research last summer and has moved to Amarillo since January of this year. In addition to his thesis research, he is also learning how to make wheat double haploid and how to screen Caspar markers linked to 30 genes or QTL. He is planning to graduate this fall. Mustafa, please take it over.
Thanks so much for kind introduction, Dr. Liu. Do you see my screen?
Yes, we can see it.
Okay. Hi, everyone. I will be presenting my master research. It is a QTL mapping for yield and yield-related traits in two wheat cultivars, TAM113 and Gallagher. Basically, I will talk about the introduction, material method, result, and also conclusion. So in the beginning, I will give some brief introduction about the wheat. Wheat is one of the most stable crops. It's primary source of carbohydrates, also proteins, and some nutrients. We can see some statistic in the table. In the bottom side, we can see the wheat production. Wheat production has increased last over 60 years, but production harvested area stayed same, remained same, and we can see also on the right side human population. It also increased 3 billion to 7 billion, and it's gonna be the 9 billion in 2050, and there will be a gap between the production and demand, so we should find a way, as the plant breeding is one of the best way to decrease this demand, to meet this demand. Grain yield is complex traits which is influenced by multiple genetic loci and other traits. Here just literature to understand the importance of yield component. For example, yield increase attributed to heading date and high 1,000 kernel weight in Mexico. In another example, increase in grain yield attributed to high grain yield number per spike and 1,000 kernel weight also in China. Our objectives here to analyze the different yield parameters in TAM113 and Gallagher population in different Texas environment, and also detect the single nucleotide polymorphisms and stable QTLs associated with yield and yield-related traits in recombinant inbred lines grown under different environments. When we came to our material method section, we have 191 recombinant inbred line from TAM113 and Gallagher. TAM113 is released by Texas A&M AgriLife Research in 2010. It has a good yield potential, also a drought tolerance. Gallagher was released by Oklahoma Agricultural Experimental Station in 2012, and it is an early maturing variety. It has a good resistance for disease and some insects, also some virus. This slide shows the summary of my experiment, and we have 3 locations across Texas. Those are Bushland, McGregor, Coal Station, and we planted our experiment using alpha lattice experimental design with two replication in 3 years. We can see the environmental codes in the last column. We used the abbreviation of location and year when we describe the location. And also we can see the plant number. Normally we have 400, but in 2019, we didn't have enough seed, and that's why we have 274. In this year, we also increased the seed for next year. Also, I have one more location in 2021. So we will harvest this summer and we will get the data also. Data collection, we have several phenotypic data. First one is the grain yield. We got grain yield data from a combined harvester in 5 environments. We record the heading date according to days from sowing time to at least 50% of heading stage. The last one in that page is the plant height data, as we record this one as a centimeter from base of plant to top of spike with excluding ounce. In yield components, we cut randomly half meter representing inner row from each plot in order to get data for a total biomass yield, biomass grain yield, a kernel per spike and 1,000 kernel weight, and also number of spike and enough seed sample for some seed traits. After we cut the plant, we put them into air dry room, 3 days for 60 Celsius degree to get more accurate data. And then we measure the all data. For data collection, seed-related traits, we use the grain scanner software because it saves time and it is also cost-effective. So we took nearly 10 grams of seed and we put them in the scanner. And here the one point we should be careful about the seed. We should separate them, otherwise the software will count 2 seed as a 1 seed and it can change the means. Software give us seed area, seed parameter, kernel length, kernel width, and 1,000 kernel weight. Software counts seeds number on the picture, and we use this number to divide the seed weight, and then we can get the single kernel weight. We can use it to calculate 1,000 kernel weight. Also, we use the single kernel characterization system. We put around 15 gram seed sample, and machine takes 300 seed to measure the single kernel weight, kernel diameter, and also kernel hardness. In statistical analysis, we use the METAR software to get the Blues values of individual environment and across environment. And then we calculate also variance components and heritability. All factors like environment, genotype considered as random effect. Additionally, we used RStudio and JUMP software to remove the outlier to get also to get the figures. On the bottom, we can see the statistical model for a single environment and multi-environment we used in our analysis. For genotyping, we send all 191 recombinant inbred lines and parents to Texas AgriLife Genomic and Bioinformatics Service at College Station. They used a double digested RATSIC protocol. After we sequenced, we had 126,000 over 126,000 SNPs marker. We decreased this number first, removing more than 20% missing data, and also we used the binding function of ICIM mapping to remove the reductant markers. Lastly, we removed the double crossovers by in Excel. Eventually, we had over 8,000 markers. It was the most time-consuming part of my project. On the bottom, we can see the marker distribution by genome, and we can see marker distribution by chromosome. Genome A and genome B has a more marker than genome D. Also, chromosome 2, 4, and 7, relatively higher marker number than others. Over 8,000 SMP marker were used to create a 25 linkage map using Join Map software. And then we mostly use the ICIM mapping to first one, as I mentioned before, the binding function to remove the reductant markers. I mean, we delete one of our duplicate markers, and then other function is the BIP. It gives us the QTL additive effect, and the other one is the MET, is the multi-environment analysis, the analyze the across environment, and it gives us the additive by environment, and also give us the epistasis by environment. We got the QTLs, our QTLs using integrated composite interval mapping function for additive effect. The permutation number is the 1000 to identify the lot score threshold to declaring the significant QTLs. The probability was the 0.05. So I will talk about our result in the next slide. Here we can see in that slide the variance components, heritability, mean performance for all traits. We found all traits significant for genotype except the head weight and the biomass grain yield. For genotype by environment interaction, all traits are significant at a 0.01 significance level except the head weight, and also perimeter is significant at 0.05 significance level. Environmental variance is also show the almost the same things. All traits significant except the seed traits. If we look at the heritability column in the last one, we can see the most of traits shows the high heritability, but head weight and biomass grain yield have a low heritability, and a spike per square meter has moderate heritability. On the correlation table, we can see the many significant correlation between traits. So is the most important ones is the dark colors is the high positive significant correlation. In the seed traits has a more significant correlation with each other. If you look the yield also has a significant positive correlation. Most of traits except heading date, plant height, and spike per square meter. Is the correlation between heading date and yield is negative significant. Is the R square is the 62%. If you look at the left side of table, we can see the scatter plugs, and in the seed area, like is the red color, here of red color, so we can see the many linear model. Here we can see box plot for yield across different environment. 2019 was irrigated. That environment showed the highest performance. The other locations were under rain-fed conditions. We can see the GG biplot on the right side of table slide. For environment, we can say we have two mega environment. Those are the South Texas and North Texas. We have multiple consistent and pleiotropic QTLs for yield and yield component in different chromosomes. Before I start to talk about the QTL, I should explain the some terms. For example, the consistent QTL means the QTL formed more than one environment, or one environment and the across environment. On the bottom of page, you can see how we give the QTL name. First one is the trait name, and then Texas A&M, and then chromosome. And the last one is the mega-base pairs of the synapse. So this figure is the composite interval mapping for additive effect for grain yield. We can see some peak locations. Those are indicate our QTLs. On the figure, we can see the red color, which means the additive effect, and the green color means the additive by environmental effect. Also, bottom figure tell us which parent contribute to our traits. If it is minus, which means the favorable allele comes from the Gallagher. If it's positive, is the allele comes from TOM113. We have many consistent yield QTL, but I'm not gonna talk about all of them. I will go through some major QTLs. For example, the QTL on 1B, which explained with 17.1% phenotypic variation and 13.1 additive effect. Also, we can see it has a high lot score. It's over 20. Another major QTL on 2D at 37 megabase pairs, which explained with 28% PVE value, which means the phenotypic variation explained. I marked 2 more yield QTL on that page. We can see some additive by environmental effect on chromosome 4A and 7D. For yield component, we have several QTL, but not many QTL for the spike per square meter. First QTL for a spike per square meter on 1A at 45 megabase pairs. It explained with 7.9% phenotypic variation explained value, and additive effect is pretty high, and allele comes from the TOM113. Another one is on 5A. The loss score is higher than the previous one. For 1000 canal way, I marked 3 QTL. Actually, we have more, but at the time may not be enough to talk all this QTL. So we have multiple QTL by additive effect. QTL on 2D, it is also sharing the same location with the major photoperiod gene, PPD1. The PV value is over 16%. Additive is small. The other QTL is on the 7D. The PV is 12.8. And the allele comes from TOM113. For heading date, we can see the one major QTL on 2D. As I mentioned before, it co-localized with a PPD1. It explained with almost 60% phenotypic variation. The additive effect is 3.5. Is TOM113 allele increased the traits. Another one is on 7D. Additive by environmental effect is very high in here. For plant high, we detect the 11 consistent QTL, and we can see also additive by environmental effect is much higher than additive effect in some point. So plant high QTLs affected by environmental effect. The QTL on 2D explained with 15% for PVE value, and additive effect is 2.1. We can see multiple QTL on 4B. One of them is on the 614 megabase pairs and explained with 8.2 PVE value. We have many QTL also for seed area, kernel length, kernel width, parameter, but I will give just one example for each of them. For here, our QTL is on 2D, and the PVE value is 19.8. The additive is very high, very low. And another one is kernel length. Our major QTL is on the 5B. It is explained with the 7.7 PVE, and additive effect is also low. The another one is on 6A. The PVE value is 12.9. The loss score is almost 8. Again, the same region has a QTL for kernel width, which is explained with 17.4, and the loss score is also high. Is the Gallagher allele contribute for this trait. Last QTL for a parameter, and the parameter and kernel length QTL is in the similar position. the QTL on 6A has a 11% PV value, and Gallagher, also Gallagher increased this trait. In that page, we can see our pleiotropic QTLs. The pleiotropic QTL means the one QTL region affect more than one traits. Before I start to talk about the figure, I should explain the colors. In the box, we can see the trait names. Those are different colors. If the trait name is purple, which means the Gallagher allele increased trait. If it is light blue, the alleles comes from the TAM113. We have many pleiotropic QTL. For an example, the QTL region on 1A at 11 megabase pairs affected two traits, biomass weight, and yield. And it is also detected in 20 college station and across environment analysis. So we have 4 more pleiotropic QTL in that page. So the QTL on 2D, the neighboring each other very close, is the first one is on 2D at 34 megabase pairs, affect the multiple traits like heading date, kernel diameter, kernel hardness index, perimeter, 1000 kernel weight, yield, and plan height. The other one is on the 2D in 37 megabase pairs positions. Also control the multiple traits, area, biomass, kernel weight, perimeter, and yield. This QTL is detected in multiple environment, 19 BSP and 20 EMN, 19 McGregor, and also the across environment. The QTL region in that page, the QTL region on 5B associated with kernel width, kernel length, perimeter, and also it detected in multiple environment, 2019 BSP, 20 EMN, and across environment. The donor parent is the TAM113, gave the favorable allele for those traits. Here, one of our QTL region on 7D associated multiple traits also is detected in multiple environment, 19 BSP, 19 McGregor, 20 college station, and across environment. So another one is on the 60. There is a 3 pleiotropic QTL region. All alleles comes from TAM113. So for epistatic interaction, we have total 168 epistatic interaction for grain yield. One of our consistent QTL is involved with this epistatic interaction, with overall lot score is 5 to 12.2. If we increase our lot score, the interaction number will drop to 18. So we have more interaction for other traits, but other major QTLs were not involved, so I didn't put them. Lastly, in conclusion, we have total 37 consistent and 22 pleiotropic QTL for yield and yield components. 18 QTL are common between consistent and pleiotropic QTLs. So those are made a higher significant level, significant additive effect on 2B, 2D, 5B, and 7D. One of our yield QTL on 1B overlapped with the previous finding by Young. Also, the major QTL, which is located at 34 megabase pairs on 2D, it is co-localized with PPD1 photoperiod gene. Lastly, we have found a QTL region on 7D at 67 megabase pairs for yield and grain related traits, which is also the same location in the previous QTL study. from Term 1 12 and Term 1 11 in Texas A&M University. This study is not complete. We have one more environment, and we will add also few more traits to our analysis. For acknowledgement part, I would like to thank all my committee members. They helped me a lot in my research. My chair, Dr. Ibrahim, my co-chair, Dr. Leo, and committee members, Dr. Roth, Dr. Malla, Dr. Okumata, Dr. Julian. Also with genetic from Amarillo, Dr. Yu, Zan, Mehmet, Kele, Casey, and undergrad student from the West Texas A&M University. They helped me in the field when we harvesting our population. I would like to thank the wheat breeding Amarillo, wheat breeding College Station, Dr. Opena, Dr. Ryan, Dr. Harhe, and Ellen, and also physiology in Amarillo, Dr. Shu and Kirk. Special thanks goes to Dr. Xinjiang Wang and Dr. Chu, Dr. Shasha, and Alperadak. So lastly, I would like to thank the Ministry of Agriculture from Turkey, the providing funding for my master. Also Texas A&M AgriLife Research and USDA provided support in my research. Thank you for listening. So now I can take the questions and comments.
Good job, Mustafa. Very nice work. Questions?
I have one question, Mustafa.
Yes.
You kind of confirmed a known hypothesis, right? That you would detect yield QTLs and there'd be way too many. So I want you to boil this information down for me and tell me how you're going to use this information. Is there any utility in it? You have too many to breed.
Can you repeat the question, please?
Okay, you confirmed a known hypothesis, something we already know. There's a lot of QTLs that control yield. So now what are you going to do with this information?
So you mean what will we do with this yield QTLs?
Are you going to use all these markers to affect breeding for yield, improved yield? Are you going to?
So we used over 8,000 markers. If your question is that.
I know you use a lot of markers, but you got a lot of positive QTLs for yield. So how are you going to use that for improving breeding for yield?
So, okay, I understood. So is the yield is not only yield, there is some other components. We cannot use the just yield, and we have to also dissect the yield components. So we have more in the future. Maybe we can clone them, and maybe we can, if we have a good QTL, we can make some marker for them. So more generally, we want to help also, we want to help the breeding programs with this QTL. So.
How's that, Dirk?
The compounding still sounding pretty good, I guess. I don't know. I'm not convinced how you're gonna boil all these QTLs down into something useful. So.
All right. Moving on. Any other questions?
Okay.
Time for one more question. Why are there so few markers on genome D?
So genome D has less markers because the D genome comes from the Aegilops tausha. So I saw with the previous studies, D genome always has less markers, and we can say D genome is very conservative. We don't see a lot of polymorphisms. Maybe it's related with the evaluation. So the genome has a joint is 8,000 years ago. So that's why it has a less polymorphism.
Okay.
I think.
Shuyu, go ahead.
Yeah, Scott, just a quick comment for the question from Dirk. We know the reason we are doing this because we try to dissect the yield component and some thick traits. From the analysis, we can tell some QTL has more or significant additive effect. That is, those QTL are our focus because breeder can fix the additive effect. And we also knew other QTL has larger interaction between QTL and environment. So those we kind of exclude in the consideration for the next step. So that is one part. I think that. So we will focus mainly for those QTL which consistent or appeared multiple environment and significant with additive effect. Then that's for our next step. Thank you.
Well, it was very nice work.